much. I'd just like to start by acknowledging three key collaborators on the work I'm going to describe. Drew Hanneman from Ichor Medical Systems, James Moon from Walter Reed Clinical Trials Unit, and Jay Hooper from USAMRIID. Viruses in the hantavirus genus of the family Bunyaviridae have been found worldwide, more than 50 species of rodents, shrews, moles, and bats. All hantaviruses known to be pathogenic from humans, however, have been detected only in rodents. And these Category A pathogens persistently infect the rodents, and they're transmitted in aerosols of the rodent's urine, feces, and saliva. They can cause two human diseases, hemorrhagic fever with renal syndrome, or HFRS, hantavirus pulmonary syndrome, HPS. Old World rodents carry the viruses that cause HFRS. New World rodents carry the viruses that cause HPS. And although these are pathogenic for people, they don't cause any harm to their rodent hosts. And this is likely because they've co-evolved with their rodent hosts for what's been postulated to be more than 100 million years. And the coevolution is reflected in their phylogenies, that genetically related hantaviruses are carried by genetically related rodents. So for this clade of hantaviruses, they're all carried by rodents in the Murinae subfamily, this clade in the Arvicolinae subfamily, and then these in the Sigmodontinae. The tight association of these viruses and their rodents also dictates where the diseases are found. Because New World rodents carry viruses causing HPS, HPS is found only in the New World. Likewise, Old World rodents carrying HFRS, causing hantaviruses, so HFRS is found mostly, but not entirely, in the Old World. The focus of our vaccine studies are the four hantaviruses that cause HFRS, and that is Hantaan and Seoul virus in Asia, mostly Puumala and Dobrava viruses in Europe and Scandinavia, although there is some Seoul virus as well. We chose the DNA vaccine approach because it has a number of desirable characteristics for our types of vaccines. They're easy to manufacture, so you can quickly design and produce them in response to emerging or genetically engineered threats, and DNA has well-established and approved manufacturing procedures. Also, they're safe. The plasmids are replication defective, not transmissible to person or into the environment. No problem with pre-existing immunity with the DNA vaccine, and it's a flexible platform, so you can mix together more than one plasmid to get multi-agent vaccines. You can deliver them by a variety of methods, and we've tested many different methods. The two that we've gone into the clinic with so far are gene gun and electroporation. So hantaviruses are medium-sized enveloped viruses, about 100 nanometers. They have three segments of single-strand negative sense RNA. The large or L segment encodes the polymerase, medium segment, the envelope glycoproteins, GN and GC, and the small segment, the nucleocapsid protein. And it's known that the primary correlate of protective immunity are neutralizing antibodies to GN and GC. And for that reason, we have concentrated on the medium M segments. We cloned the M segment of Hantaan virus into a plasmid downstream of the cytomegalovirus promoter and intron A, upstream of bovine growth hormone, polyadenylation transcription termination signal, and then there's a kanamycin resistance gene for selection in bacteria. So when this plasmid is introduced into mammalian cells, the CMV promoter is recognized by host cell polymerase, and the DNA is transcribed, then translated into to yield the GN and GC glycoproteins shown there, precipitated with monoclonal antibodies, and compared to authentic Hantaan virus proteins precipitated with polyclonal antibodies. So there is no animal model for disease for hemorrhagic fever with renal syndrome, but we've developed a hamster infection model that we've used extensively. And in this model, we vaccinate, then look for neutralizing antibodies, and after challenge, we look for antibodies to the nucleocapsid protein. Since that isn't a component of the vaccine, if we have antibodies to nucleocapsid, we know they were not protected. And our hamster work's been published for a long time, so I'm just summarizing. We found that the Hantaan vaccine elicits neutralizing antibodies and protects from infection with Hantaan virus and also protects against Seoul and Dobrava virus infections. It does not protect against Puumala virus infection. So again, looking at the phylogeny, you can see the reasons for that, in that Hantaan, Seoul, and Dobrava are 77% identical in their GN and GC amino acids and have some cross-neutralizations. Puumala is only 53% identical with little or no cross-neutralization. So our vaccine, we knew we needed two components, one expressing the M segment of Hantaan and the other of Puumala. And as I said, one of the advantages of a DNA vaccine is you can mix together several to get multi-agent vaccines. But what we found is when we mixed Hantaan and Puumala plasmids together, we were getting neutralizing antibodies only to Puumala. We couldn't overcome this even when we did a higher ratio of Hantaan to Puumala, as high as 10-to-1. So for our first clinical study, which was the gene gun study, we gave both of them to the same individuals but administered them separately, for which we had no interference issues. So again, this was published in 2012, and I'm just going to summarize the results. In this phase 1 study, we had 3 vaccine groups of 9 subjects. One group got Hantaan, one got Puumala, and one got both DNAs. Vaccines, there were no adverse events, well tolerated and immunogenic. Some of the volunteers produced high levels of neutralizing antibodies, but the overall seroconversion rate was just under 50%. It was just over 50% for those that got both vaccines, and that wasn't good enough. So we decided we probably need a better delivery method, which is when we turned to electroporation. And these studies were done in collaboration with Ichor Medical Systems. With electroporation, the DNA is injected in the muscles through a central needle, and then outer needles serve as electrodes to provide a very quick voltage pulse. And it's believed to create temporary pores in the membranes of cells, allowing the DNA to be taken up better than it would be with just a needle injection. We first did a preclinical safety tox study in rabbits before going into the clinic. And we used rabbits for this rather than hamsters because they have a large enough muscle mass so we can use the human delivery device for the preclinical tox studies. And they were safe in the rabbits by all of the criteria that we measured. And looking at neutralizing antibodies in those rabbits after 3 vaccinations, we saw that those that got Puumala or Puumala mixed with Hantaan had high levels of neutralizing antibodies. Mean titers were about the same. But for Hantaan, when given Hantaan alone, they had higher neutralizing antibody levels than when mixed with Puumala. So the interference we saw in hamsters was still occurring in rabbits, but unlike hamsters, it wasn't absolute. Some of these rabbits are getting pretty high levels of neutralizing antibodies. We decided to go forward with our study in humans using the mixed vaccine. This would allow us, again, we did it just like the DNA study with gene gun with 3 groups, Hantaan, Puumala, and mixed Hantaan and Puumala. And this allowed us to determine if electroporation would improve seroconversion rate as compared to gene gun, but also to determine if interference also occurs in humans, as it did in hamsters completely and rabbits somewhat. So to skip just to the results here, we had no serious adverse events, and these are the neutralizing antibodies, which is what we were most interested in. These are for the Hantaan vaccine. People were given 2 milligrams of DNA 3 times at 4-week intervals by intramuscular electroporation. 2 people dropped out of the study after receiving 2 doses for non-study related reasons. One was a bicycle accident, and the other was somebody had a personal problem. But anyway, we included those 2 people in the orange boxes in the results here. So including them, we had 7 out of 11, or 64%, that developed neutralizing antibodies to Hantaan virus. For Puumala, we had 6 out of 8, or 75%, developed neutralizing antibodies, and actually the 9th individual, shown here with the dotted line, also developed neutralizing antibodies, but for some reason at day 0, this person had a 1 to 20 neutralizing antibody. It was probably either nonspecific or cross-reactive. So actually it's 7 out of 9, or 78%. If we look at those that got both vaccines, for Puumala, this again is the mixed vaccine, same dosage, 1 milligram of each DNA, 3 times 4-week intervals. We had 7 out of 9, or 78%, developed neutralizing antibodies to Puumala. For Hantaan, there were only 3 out of 9, or 33%. And interestingly, the ones that did develop neutralizing antibodies to Hantaan had the high levels to Puumala as well. So this is kind of like what we saw in the rabbits. Some people would develop high levels of neutralizing antibodies to both, but not everybody. So to summarize what I've said so far, the Hantaan and Puumala DNA vaccines delivered by IM electroporation were safe and immunogenic in the phase 1 study. There continued to be interference with the Hantaan vaccine to some extent, but luckily while that study was ongoing, we were able to solve that issue of interference, and all it took was optimizing the gene for the Hantaan virus vaccine to codon optimize it for Homo sapiens, removing cryptic splice sites, highlight regions of GC content, and some of the other things that GeneArts does. And once we did that and tested it in hamsters, see that it actually gives higher levels of neutralizing antibodies than the non-optimized version. And when it's mixed with Puumala, we're getting neutralizing antibodies now to both Hantaan and Puumala. So we've moved on using the 2 optimized plasmids in a phase 2A dose ranging study. And in this study, we're testing 2 doses, 0 and 56 days in comparison to what we'd done earlier, 3 doses at 4-week intervals. And we're testing 1 milligram or 2 milligrams of DNA in groups of 30 individuals. And for all of the groups, we have an option of a 6-month booster if we choose to do that. And right now, this study is ongoing at Walter Reed, and we vaccinated about 30 people so far. We also have a NIH contract to compare these mixed DNA vaccines using intramuscular compared to intradermal electroporation. And that study is funded, but it's stalled right now in the manufacturing of the DNA because the company's having a hard time getting a high enough concentration for the intradermal electroporation. But we hope to be able to start that within the next 6 months as well. So I'd just like to conclude then with normal disclaimers, showing a picture of our new USAMRIID over here, which we hope to be in about 2 years. So thank you for your attention. Any questions?
What are you doing anything about the isotype of particular antibodies you're inducing the DNA vaccine?
In the humans, we've done it in animal models, but in humans, we have that on the list to do, but it isn't done yet.
Do you find it a bit surprising that you generate such decent antibody responses with an unoptimized genetic material through a DNA vaccine? Because most people don't see that.
No, all of our DNA vaccine studies, we've done clinical studies with Venezuelan equine encephalitis virus, and we get very high levels of neutralizing antibodies as well. Even with our Ebola DNA vaccine, we're getting neutralizing antibodies, which I think it has to do with the delivery methods more so than the DNA.
Very nice talk. Can you say us more about your DNA Ebola vaccine?
Yeah, I just did that talk in Montreal. We did a DNA vaccine for 4 filoviruses: Ebola, Zaire, Sudan, Marburg, Musoki, and Rabin, and did non-human primate studies where we vaccinated with individual or the combination, and then challenged with either Zaire or with Marburg, Musoki. And we found that the Musoki vaccine protected all of the monkeys, whether given alone or in combination. For Ebola, the DNA vaccine protected all but one of the monkeys when given alone, but when mixed with the others, it only protected one. So it's a whole talk, but those are the results.
Some studies have demonstrated that heterologous prime boost immunization protocols using such as DNA and proteins, DNA and recombinant virus have improved the immune response. Have you tried heterologous prime boost immunization protocols?
Not with these, we haven't, because we figure why do it when we're already getting such high neutralizing antibodies? Why develop 2 vaccines and go through 2 regulatory hurdles if you don't need to? So no.
What you're getting with this mix, the dual vaccine, if you like, and putting them in together at the same place, reminds me of what's happening with dengue virus, where you only get sort of immunity to 3 out of 4 types when you put all 4 of them in together. Have the two situations got something to learn from one another that they can both improve?
I don't know. I really don't know what was the interference issue before it was codon optimized to start with. Something was happening that destroyed the Hantaan messenger RNA. But by codon optimized, because the Puumala was always optimized, 'cause it wouldn't express until it was optimized. The Hantaan one was just fine. It expressed well, it was immunogenic, so we didn't think we needed to do that. But once we did, there's no issues anymore. I think it's a messenger RNA issue.
Thank you.